Good afternoon, everyone. Welcome again to the second, seminar from the story speaker, Professor Spencer from University of California, University of Missouri, Columbia. So we are very glad, Dr. Spencer again spent past 2 days with us. So we had really lovely discussion about IVF, other topics. So today it's a more of a scientific seminar. And then, so I try to give a very short of introduction if some of you guys was not here yesterday. So Professor Spencer is a curated distinguished professor, and then also he's a vice chancellor for research from University of Missouri, Columbia. And then he's a founding director for this Next Gen Position Health Group, and also founding director of the interdisciplinary of the reproductive and health. And then he's a member of National Academy of Science. He has received numerous of the awards, just coming for a couple of these research awards from, Association for the Animal Science, Society of the Reproductive Societies, and then he has published, 270 peer-reviewed papers with H-index of 120. It's very, very impressive, very productive. Without further ado, I give to you, Tom.
Right, thank you very much. Right, that should be on now. I'm a little less tethered today, which is good. So I really have appreciated the invitation here. Everybody has really shown some really gracious hospitality, particularly Wei Zhen. Thank you very much. And I've really enjoyed meeting many of the people that are in the audience today and renewing some old friendships. So it's really been a great visit and to arguably a very beautiful university campus. So today I want to talk about one of my favorite topics. Remember, I am Captain Uterus. And so I want to talk specifically about something that I think now I've spent about 30 to 35 years of my life studying, which is the development and function of uterine glands. And my son, my 11-year-old son, is actually reading Homer's Odyssey in his school. And so I appropriated that because I think it's kind of very similar where, you have these vignettes that occur over time, and really certainly it's been a voyage in terms of the development and function of these important cell type in the body. So I would be remiss, I usually like to thank the people that are doing the work. So this is the members of the Spencer Kelleher Lab, Andrew Kelleher on the left. Both he and I share lab space and a lab group together, and really allows me to continue to do research while I delve into administration. I'll highlight some of the work of the former lab members, and particularly I have some collaborators both outside of Mizzou as well as inside of Mizzou that have really helped take this area of research forward. And then I've been fortunate to be funded predominantly by the National Institute of Child Health and Development for the work today in mice and humans. And I will begin with some work that was originally funded by the National Institute of Food and Agriculture. So we're going to talk about the uterus today. I know most of you, except for maybe Richard, understand the uterus, but this is the human uterus. The function of it, our conception, is a conduit for sperm to reach the oviduct for fertilization of the ovulated egg. It is the preferred site in humans and the only site for pregnancy in domestic animals, for implantation of the embryo formation of a conceptus, development of the placenta is, of course, critical for nutrition of that fetus. And then ultimately, the outer smooth muscle layer or myometrium is responsible for labor and delivery of the baby at term. In the study of the uterus, actually, been a lot of people that have studied the uterus throughout time. Originally, you can go back to Galen, who first wrote about functions of the uterus. Obviously, there was a period of dark ages, and then during the enlightening period of Leonardo da Vinci, where we first really got an insight into what we thought was the human body. So you can see pictures that he published. But you'll note here, this is a human fetus within the uterus. The uterus is generally correct, but the placental structure is that of a deer because it has about 6 placentomes. And that's because vivisection was essentially not allowed at the time. And so because they would hunt animals, and a lot of them were pregnant, and he would look at pregnant deer, and he surmised that, oh, the human placenta must be the same. He generally got it correct, and I think he did pretty good for the time in terms of what he could explore. So specifically, we're going to focus on the endometrium. And the endometrium is replete with a number of different cell types. And here, this is a famous section of the pregnant uterus that was originally published in 1932. And actually, I have an original copy of the book, and you can pull out the page. And essentially, it's a histological slide that then was reproduced by hand and pseudo-colored, which is amazing for 1932, where you can see the conceptus, the developing placenta in purple, and then you have this conceptus on a large bed of glands of the uterus, supported by stroma. And you'll note that the glands of the uterus are larger at the implantation site, and they are secreting factors or milk that are directly delivered to the intervillous spaces for nutrition of the developing fetus as well as the placenta. And so the predominant cell types are the luminal epithelium that coordinates attachment of the embryo for implantation and also acquires receptivity for this event, primarily under the control of estrogen and progesterone. Then you have the glandular epithelium, which we're going to talk a lot about today. These glands either selectively transport or synthesize and secrete proteins and other factors to nourish the embryo that regulate development of the placenta. And they also produce factors that regulate development of the decidua. So the stromal cells support the epithelium. These stromal cells actually are instructive for development and function of the different types of epithelia. And they also differentiate into specialized decidual cells that directly regulate placental development, as well as maternal immune cells that orchestrate placental development. And then it is replete with blood vessels that provide essential nutrients for development of the fetus. And I think it's one of the most interesting organs because in the adult, it is one of the only organs I know that in an adult pubertal woman continue to regenerate each and every 30 days, where you have sloughing of the upper functionalis endometrium during menstruation, regrowth during the follicular phase, transition under progesterone after ovulation and formation of the corpus luteum, and undergoes initial decidualization in preparation for embryo implantation. So why would you want to focus on glands? like, why do you want to commit your life to trying to understand the biology of these interesting facets? Well, number one is they do regenerate every menstrual cycle. And we know that they synthesize, secrete, and transport a variety of substances, both macro and micronutrients, into the uterine lumen. And some more work today that I'll talk about, as well as a number of other investigators, we know that they are essential for embryo survival and implantation. They contribute, particularly during the first trimester of pregnancy in humans, to conceptus and fetus growth, as well as formation and development of the placenta. In addition, they are involved in literally almost every women's reproductive health problem or disease. So their dysfunction can contribute to infertility. They are a dominant cell type in endometriosis, where you have growth of the endometrium in the peritoneal cavity outside of the uterus, which is very painful and very problematic in about 10 to 20% of women worldwide. If they overgrow into the myometrium, this causes actually quite high levels of pelvic pain and is associated with infertility. It's a condition called adenomyosis. And then, of course, pretty much one of the dominant cancers of women is endometrial adenocarcinoma, and that is predominantly benign growth of cells of the glandular epithelium to form cancer. And they also hold a lot of promise in terms of precision medicine approaches. So one would be biomarkers of fertility, particularly important, as we talked about yesterday in length, in terms of IVF programs, and also thin endometrium, or there are conditions like Asherman syndrome where you don't essentially have an endometrium. Stem cell therapy approaches would be particularly important in terms of precision medicine approach to solve certain reproductive health problems. And I just think that they're fascinating. This is the duct of a gland on the surface of the lumen. So this is luminal epithelium, and this is the duct of the gland. And you can see here in this electron micrograph, tufts of ciliated epithelium and these actual large secretions that are emanating from these ducts and bathing the uterine lumen where the developing embryo and conceptus would be located. And this is a three-dimensional reconstruction of the glandular architecture of the uterus. You can see these rhizome-sharing glands. So these glands, the ducts empty into the uterine lumen, but they have this coiled and slightly branched architecture that then in the basalis endometrium has a completely different architecture. And of course, that's where the stem cells reside for regrowth of the functionalis endometrium each and every menstrual cycle. So if you look at textbooks, and actually this is from a review article by Danny Schust and Earl Norwitz in the New England Journal of Medicine in 2001, basically this is what you're taught in medical school about human implantation and pregnancy establishment, and what pretty much everybody thinks that they know. So you have fertilization in the oviduct, rather quick entry of the morula into the uterus by day 4, formation of a blastocyst, attachment, and then initial nidation of the embryo by day 8 or day 9. So this embryo attaches to the luminal epithelium. It was thought at the time that the glands secrete factors that nourish the embryo, but once it started the implantation process, essentially the only important cell type was the stroma that was undergoing this decidual cell differentiation event. And indeed, it was thought that essentially as the placenta developed and early embryogenesis, essentially this was all under the regulation into 2 different cell types with within the stroma. So stroma and decidual cells, as well as a number of natural killer cells called uterine NK cells, and then nutrition was provided by the spiral arterioles. But it turned out that this was challenged initially by histology of the endometrium. And this is actually some work from Alan Enders, who was a faculty member here at the University of California, Davis. I knew him when I was a young graduate student. It was always a pleasure to meet with him. He had a fascinating knowledge of pretty much the uterus and endometrium of almost any species that you would want to discuss with him. And if you look at this trophoblast plate stage, this is actually a non-human primate implantation site, but you'll note that this is roughly probably around day 10 to 12 human equivalent. Well, the embryo hasn't magically nidated and destroyed the endometrium. It's right here. This is a syncytiotrophoblast, an inner cell mass. This is the cell type that produces chorionic gonadotropin, which is the pregnancy recognition hormone. These are actually immune cells and then some decidualizing stromal cells. But here is the glandular epithelium, and actually some work from both Alan Enders as well as Bertel Hopperts in Belgium found that there are some implantation sites where the embryo actually directly nidates into the opening of a uterine gland. And so this really challenged the dogma that glands were not important because they maintained their functionality, as the embryo didn't magically nidate like a mouse embryo, but slowly began to interact with the endometrium as it began to form the placenta, and you had early stages of embryogenesis. And some really pioneering work from Graham Burton at Cambridge. They had a series of samples that were collected in the early 1900s called the Boyd collection. And essentially they paid women to get pregnant, and then they would hysterectomize them, which I'm pretty sure would never pass an IRB right now. But there was a collection both at Harvard as well as in Cambridge where you could actually look at early implantation stage samples from humans. You'll note here, this is day 10, and what are the features here? Well, number one is this is the embryo and the developing placenta, but you'll see here these large numbers of uterine glands, and directly under the implantation site, you have glands that originally were thought to be cancerous, but they're actually just hypertrophic because there are factors from the placenta like chorionic gonadotropin and placental lactogen that are secreted by the syncytiotrophoblast and are causing hypertrophy of those glands so they can secrete factors that are delivered to the developing intervillous spaces of the placenta. And here we've already discussed the famous Faulkner embryo, but look at the 6-week implantation site. So this is the developing placenta on this huge bed of functional uterine glands. And so he developed this hypothesis that was published in a series of papers in 2010s and early 20 that basically established the concept that uterine glands are functional throughout the first trimester of human pregnancy and would secrete factors that could directly affect development of the conceptus and the placenta because those factors were in the developing intervillous spaces of the placenta. And about the same time, it was recognized that these spiral arterioles were actually plugged during the first trimester of pregnancy and were then unplugged during the transition to the second trimester, where you would get direct provision of blood to the developed placenta. And so this really revolutionized some of the concepts of what we thought about early pregnancy in humans. So now I want to transition and really talk about what do we know about glands and other species, and then we'll kind of concentrate on gland development. So pigs are pretty remarkable when you think about it. They have true non-invasive implantation with a true epithelial chorial placenta. There's no erosion of the uterine epithelium and invasion into the endometrium. And when you peel away the placenta from the uterus, number one, you can see the lumen of the uterus. These gland openings are just like what I showed you earlier, where you have secretions being transported into the uterine lumen. But then when you look at the surface of the placenta, I think this actually looks like the mouth of a lamprey. And this is a specialized structure on the placenta termed a areolae. And these structures form over the opening of a duct of the uterine glands. And this is shown histologically here. This is some work from Greg Johnson at Texas A&M. You just note these are the glands in the endometrium. And this is this areolae, and you can see that these secretions are directly delivered into this space. And Fuller-Baser showed in the 1970s that if you take piglets at term and you save the placenta and you weigh the piglet and you count the number of areolae, that the number of areolae correlate to the birth weight of the fetus. And so glands, particularly in a non-invasive implantation, are critical throughout pregnancy to support development and growth of the placenta, particularly through these areolae structures. What about ruminants? Well, sheep, cattle, goats, and deer, they have a limited erosion and penetration and invasion of the endometrium. They have these intercaruncular areas of the endometrium that contain glands, and then they have these caruncular areas that are aglandular. Eventually, you'll have cotyledons of the placenta interdigitate with these maternal caruncles to form these placentomes. And this is a site where you get hematotrophic or blood-borne nutrition. But in these intercotyledonary areas, they have a true epithelial chorial placenta that develops these areolae. And so here I dissected off, this is an early placenta of sheep. And so this is the developing, I peeled away the cotyledon that would become a placentome. But you'll note this milky substance, this is actually uterine milk that's produced by the glands. And if you make a unilateral pregnancy where you tie off one uterine horn, so you only have the fetus in one uterine horn after you mate them, this actually horn will fill up full of uterine secretions, and you can actually empty it into a 50 mil conical tube and collect about 40 or 50 mils of uterine milk. It's like phenomenal. And so for many years, we actually used sheep as a model system. And we looked at the gland life cycle in sheep. And so a couple of take-home messages. One, there are no glands at birth. They rapidly develop during the first and second week of life. They're essentially mature by 8 weeks of age. As the animals cycle, they have large numbers of glands in the intercaruncular areas of the endometrium. But once they undergo pregnancy, they go through a pregnancy-dependent hyperplasia followed by hypertrophy. Exactly the same that we saw under the implantation site of the early human embryo. And then after parturition, you don't need all this glandular structure, and so you go through involution, where essentially within a few months, you have a cyclic type of smaller glands, and then this cycle is repeated. And in some work that I don't really have time to go on today, we worked with a couple of colleagues at Hebrew University of Jerusalem, Ari Gertler and Alicia Gutwein, and we basically showed that there were factors from the placenta, like placental lactogen and placental growth hormone, that were responsible for this hyperplasia and hypertrophy of the glands, along with progesterone from the corpus luteum and placenta. So if you look at basically all mammalian uteri and the little bit of information that we have from humans, postnatal development of the uterus is unique because the uterus is not mature at birth. So gland development or adenogenesis is primarily or uniquely a postnatal event. Here we can see mice as well as sheep. We're using a conserved gland marker called forkhead box A2 or FOXA2. This is immunostained in red with the epithelium in green, and you'll note in mice as well as sheep, these glands begin to generate from particular cells in the undetermined luminal epithelium, and they develop rapidly over the first few weeks of life. This is initially both ovary and steroid independent, but is susceptible to programming by exposure to steroid hormones, particularly estrogens and progesterones. And we're going to talk about the progesterone-induced uterine gland knockout model. I did talk about that yesterday, but the concept of this was originated by some work from Skip Bartol at Auburn, and he reasoned that maybe it was the withdrawal from progesterone at birth that initiated development of the glands, and so you might be able to create a glandless uterus by exposing neonatal animals to progesterone after birth. And indeed, that's how we developed together the uterine gland knockout sheep model. So we exposed neonatal ewes to progestin from birth to postnatal day 56, and we found this would permanently ablate gland development. So the initial adenogenesis is a critical window of development, and like many other organs, when you bypass that, you can never go back. The differentiation of other female reproductive tract structures, as well as the ovary, is unaffected. And we also, as far as you could assess in a sheep, function of the brain and the ovary in the adult was not affected. And so we worked for a number of years in this model where we had normal control animals, 500 or 600 uterine gland cross sections per uterus. In the uterine gland knockouts, completely no glands, only areas of these caruncles. And we found that they were infertile due to recurrent early pregnancy loss. We also found that if you transferred embryos from controls into them, you could not ameliorate this defect, but if you transferred embryos developed in uterine gland knockout ewes, they would establish pregnancy in control gland-containing ewes. So it really was a uterine-based defect. And this is important because this was the first demonstration that glands in the uterus were absolutely required for pregnancy establishment. And Allison Gray, a very talented PhD student with me at Texas A&M, we did a number of different studies, and she basically found that in these uterine gland knockout ewes, you had a failure in conceptus survival and elongation after hatching and formation of the blastocyst on day 8 and day 9. And so for a number of years, we used a variety of physiological genomics approaches. Actually, when I first met Wei Zhen at Texas A&M, we were doing a lot of this work. Many of you probably will not even know what some of this was. We were doing differential display, subtractive hybridization, a lot of sequencing and blasting sequences, which was pretty painful back in the day. It's a lot easier now. And then we basically made our own custom ovine cDNA arrays, and we're looking at differences between these. And we found over the years a fascinating array of different factors that emanated from the uterine glands and the luminal epithelium that through a variety of either in vivo or in vitro studies regulated growth and development of the trophoblast. And we really discovered some kind of crazy things like endogenous retroviruses of sheep, which we studied for a number of years, and then a family member called galectin-15 that actually is secreted by the glands under the influence of progesterone. but when it's taken up by the trophoblast, it was assembled into crystals that you could actually see with a microscope in the developing placenta, which was one of our original papers we published in the National Academy of Science. And so over time, we developed a concept using this model that in the uterine lumen, the factors from the epithelium would customize the uterine luminal milieu and provide an ever-increasing source of factors to regulate growth and development and survival of the conceptus. And these factors would regulate trophoblast survival, proliferation, migration, attachment, and adhesion. And so these studies really were the foundation that the success of uterine gland development determines in a large part the embryotrophic potential and functional capacity of adult uterus. And as Norman Wessels, a very famous developmental biologist, said, it's only structure after a structure is formed that its function becomes important. And so another part of the lab for years used the neonatal U as a model system, and actually this is the first NIH grant I ever received, was using the neonatal U as a model system to study gland development. And we were looking both at extrinsic as well as intrinsic mechanisms that would regulate proliferation and differentiation of the glands. So we found factors like prolactin from the pituitary. There were factors from the ovary like activins and follistatin, and these intrinsic mechanisms that were predominantly stromal-derived growth factors that had epithelial receptors that regulated this process. So I'll share one story by Karen Carpenter in the lab and Kristen Taylor. So we looked at circulating levels of prolactin after birth. You'll notice a tremendous increase to 500 nanograms per mil during the first 2 weeks of life that correlated with the genesis of glands and expression of the prolactin receptor, both the short and long forms. And then we inhibited prolactin release from the pituitary using a well-known factor called bromocriptine mesylate, which is a dopamine D2 receptor agonist and a prolactin secretion inhibitor. So we put neonatal lambs on this bromocriptine and then looked at 8 weeks of age, and we could suppress circulating levels of prolactin. You'll note reduced numbers, particularly of these slightly branched glands. So we had about a 35% reduction in a gland number, indicating that pituitary prolactin was one of the extrinsic factors that were driving gland development. And then we asked the question, could we make a better glandular uterus? So we worked with recombinant ovine prolactin provided by our collaborators in Israel, and we gave ewe lambs induced hyperprolactinemia with injections of recombinant ovine prolactin and looked on day 14 and day 56. So this was very effective in increasing circulating levels of prolactin. And you note there's really not a tremendous increase or any effect on ductal gland invaginations, but we consistently saw about a 2-fold increase in gland number. And actually, for a number of years, we were working with a biotech company to try to increase prolificacy of ewes that were hyperprolific due to increased ovulation rate using a gene therapy approach with injection of a prolactin construct into the muscle. Unfortunately, it never actually worked, and so we moved on. And so really, this study's established really one of the first extrinsic factors where we could show that something from the actual brain or pituitary would modulate development and function of the uterine glands. And so also there's a variety of sources in humans where you have prolactin insufficiency as well as overproduction through tumors, and this could cause subfertility or infertility due to effects on endometrial regeneration in humans. So I'll show another vignette before we talk about some more recent data. where we also looked at the well-known Wnt signaling pathway that's involved in the morphogenesis of many organs. There's a canonical and a non-canonical pathway that regulates adhesion, proliferation, and migration of cells. And there's both a well-developed canonical Wnt signaling pathway that activates nuclear signaling. And there are also these factors that inhibit Wnt action by preventing the actions of Wnt or binding and activation of the receptor. And so we went through all the Wnt's in sheep. There's about 20 different family members. You'll note that some of the activators are expressed in different cell types like the luminal epithelium and glands. The receptors are present as well. But what we found is one of the inhibitors, secreted frizzle-related protein, specifically showed up in the aglandular caruncular areas. And when you really look at the ontogeny of expression, you'll note that it turns on when glands are developing in the intercaruncular areas, but only in the caruncles. And beneath the glands, at the apposition and interface of the myometrium and endometrium. And so it's just fascinating that, you know, this probably controls local Wnt morphogenetic gradients and concentrates Wnt signaling directly in the adenogenic region of the uterus, which is the intercaruncular area. And we did some studies where we're looking at effects of estrogen and progesterone to inhibit adenogenesis, and basically Kanaka Hayashi found in the lab that if you give neonatal ewes estrogen, this inhibits gland development, but is associated with the upregulation of this Wnt signaling inhibitor. And so this pathway now we know through studies in rodents, as well as through a variety of studies using endometrial epithelial organoids in vitro and 3D culture, that Wnt's are absolutely critical for many different stages of luminal and glandular epithelial development and specification. And so we developed a number of working hypotheses in the neonatal ewe where you had active and inactive areas of Wnt signaling that controlled why glands develop in the intercaruncular area, but not the aglandular caruncles. Well, unfortunately, we took the ewe about as far as we could. And at that time, there were really no mechanistic approaches where we could, let's say, manipulate signaling pathway or genes directly in the developing uterus. And so that was about the time where particularly Cree-LOX technology and the ability to create conditional mutant mice had come fully on board. And so we decided to switch model systems because science begins with a question, whereas technology begins with a task. And so we transitioned where we predominantly, and still today, use mouse genetic models for the studies of developmental biology of the uterus. And so just like we talked about the sheep, adenogenesis in the newborn mouse occurs during the first 3 weeks of life. You have initial budding of the glands on day 8 to 10, and then they begin to grow down to the myometrium, and this process is essentially complete 3 weeks after birth. And so we took the progesterone sheep model and we transitioned it to mice, where we found that if we expose C57 black pups to progesterone from day 2 to day 10 of life, this would permanently inhibit gland genesis and create an adult uterine gland knockout phenotype. And it turned out that somebody used this same type of model, Prandeep Tanwar's lab in Australia, and he basically found what we found with estrogen in the neonatal sheep, that progesterone here would inhibit canonical Wnt signaling. They're using a readout of a GFP mouse reporter. It would upregulate, in this case, an inhibitor called DICOF2 or DKK2, and this was followed by a complete disruption and downregulation of canonical Wnt signaling. So it's really kind of neat when you see something that appears to be a common mechanism across species. So our adult uterine gland knockout mice, or the PUGCO mice, basically completely lacked endometrial glands, and we used a marker called FOXA2, which is a gland-specific marker in all studied mammals. So you can see here, none in the PUGCO mice, and we also couldn't find other gland-specific genes like SPINK3. And we found through a variety of studies in these glandless mice that they have a failure in embryo implantation. So you don't see nidation in the initial stages of placentation. The embryo is sitting here in the uterine lumen. You can flush it out of the uterine lumen because it doesn't even attach to the luminal epithelium. And you also don't get stromal cell decidualization like you have in a normal pregnancy. Actually, you have inappropriate upregulation of a number of decidualization-related genes in the luminal epithelium, which is a topic for another day. So this indicated that glands not only regulated embryo implantation, but also probably influenced stromal cell decidualization. And so we did a lot of work in this area over a number of years with many talented trainees in the lab. And so kind of the working hypothesis here that we developed over a number of years is that under the influence of estrogen, these FOXA2 positive glands would secrete LIF. This LIF caused acquisition of attachment capacity of the luminal epithelium for blastocyst trabectoderm attachment adhesion, and then also influence the ability of the blastocyst to initiate stromal cell decidualization. And indeed, just like humans, the glands continue to function during early pregnancy. You can see here, these are the glands marked by FOXA2, and in this three-dimensional reconstruction by Ripple Aurora's lab at Michigan State, you'll note that these glands actually have direct ductal connections to the nidation site and the early embryo that's undergoing embryogenesis and early placentation development. So we actually transitioned from the PUGCO model to using a conditional knockout model of forkhead FOXA2 because Jaewook Jiang and Franco De Maio had found that if you conditionally delete this gene using the progesterone receptor Cree model, that you could produce a completely glandless phenotype that exhibited embryo implantation and stromal cell decidualization defects. And actually, we have studied FOXA2 and continue to study it in the lab because this is a very interesting transcription factor that's a pioneer transcription factor that can open and engage chromatin. It has conserved expressions in the glands of the human and all other studied mammalian uteri. It's also a well-known tumor suppressor gene that is mutated or downregulated during the development of endometrial cancer, as well as in eutopic endometrium that is the foundation for the development of endometriosis. And so this remains a central facet of what we're investigating in the lab. And indeed, our lab and others, we know that the loss of FOXA2, you don't get the development of glands, and this compromises pregnancy. And so our lab and others published a series of papers indicating that FOXA2 is a critical regulator of gland differentiation and adult uterine gland function, that uterine glands and their secretions coordinate on-time embryo implantation and stromal cell decidualization. And actually, a recent report from S.K. Day's lab indicates that a lot of the loss of these in the gland knockouts is due to asynchronous embryo implantation and defective decidualization. So the implications of this work are that if you have dysfunction or dysgenesis of the uterine glands, that this is going to cause problems with the early stages of implantation and placentation. And we know that in the human particularly, if you don't have on-time implantation or you have insufficient stromal cell decidualization, that this is associated with later pregnancy complications, such as preeclampsia and fetal growth retardation. So we really think that any disruption in these early glandular processes basically can cause a lot of later pregnancy complications. And this has been borne out in a number of different studies, predominantly done by Carlos Simon in Spain. So let's talk about some current research as we finish the talk today. So we're really trying to understand how FOXA2 is regulated so we can understand how it regulates as a master transcription factor uterine gland development and function. And we also have some studies where we're looking at its role in endometriosis, and there's some other labs looking at its role in the development of endometrial cancer. We have predominantly are using the mouse as a genetic model. And so we've developed a number of different Cree mouse drivers where we can do conditional knockouts. We have a new one that we haven't reported yet that will target the initial developing glands in the neonatal uterus. And we've reported last year an adult uterine gland specific, this PRSS29 Cree. And so we are using a combination of mouse genetic models as well as we are studying endometrial epithelial organoids that we generate from mice, as well as we have a biobank of human endometrial epithelium that we get endometrial biopsy samples from women, and we use these as complementary in vitro models to study the developmental biology and specification of the epithelium of the uterus. So we really focus now, and our current grant that we just received funding for really is focused on how do these glands undergo their initial genesis and morphogenesis, and really looking at mechanisms whereby the early epithelium is specified, and then how do you get glandular epithelium bifurcating from a specified luminal epithelium of the endometrium. And we're looking at intrinsic factors like FOXA2, as well as stromal epithelial interactions. And we also have some projects where we're looking at extrinsic factors from the ovary, as well as other organs and tissues in the developing neonatal mouse, and really focused on this epithelial lineage specification and bifurcation. So one of the studies we recently published in the National Academy of Science Journal is single-cell RNA sequencing of the developing uterus epithelium. And so what's really cool about the early neonatal uterus is you can trim off the oviduct and cervix and vagina, and then through a very brief enzymatic treatment, you then can, using a very fine pipette, you can remove the entire epithelium as a tube. And then we can break that down into single cells and then do 2next genomics, chromium, and Illumina sequencing, and then subsequent bioinformatic analysis. So we can completely get rid of any stromal cells, and we're only focused on the epithelium. And so we recently published this where we're looking at 1, 5, 12, and 15. This is before gland development during epithelial lineage specification, and then during glandular epithelial bifurcation. And so what I think is quite surprising is we found a number of different epithelial cell types, both in the undetermined epithelium as well as the determined luminal epithelium and the developing glands. So there's a number of different cell populations that are present in the developing uterus, and I think this is really the power of single-cell sequencing, is really trying to understand the phenotype of these cells at a molecular level. So we did some work where we kind of did a composite analysis of undetermined and determine epithelium. We followed this up by looking at factors that we think are involved in epithelial maturation and lineage specification by in situ hybridization. So here's FOXA2 in the glands. We are looking at retinoic acid signaling because the retinoic acid pathway appears to be active in the developing glands. We've also uncovered a number of different transcription factors like CYTID4, and we currently have developed FLOX models for these, are mating them with our conditional models, particularly the CXCL15 Cree. So this single-cell sequencing project really has allowed us to develop a working hypothesis framework for different cell types, like determining and proliferative progenitor populations, and then LE progenitor and specified populations, and both proliferative and stem cell and glandular epithelial progenitors, as well differentiated. And I think what's really powerful is not only can we use our conditional analysis knockout models, but also because we have these in vitro endometrial epithelial organoids that recapitulate the developmental biology, we can manipulate these rapidly in vitro and get readouts in terms of how they impact the different cells and their populations that we think are involved in these primary morphogenetic events. So the final project that I'll illuminate today is we're really focused on how does FOXA2 regulate gland development. Because remember, if we knock out FOXA2, they don't get glands. And so our single-cell sequencing experiments actually highlighted that if you look at these FOXA2 positive cells, that there actually are two different types of signatures, indicating that there might be two different glandular populations where FOXA2 is active. And then we used a really kind of cool mouse model called the FOXA2 venous fusion that we got from Heiko Lickert in Germany. And if you look at this, you can see it's only in the glands, but you'll note there's both a low as well as a high population of these FVF marked cells. That would be, this reporter is a direct readout of the amount of FOXA2 in the cells because of the way it was made. So Hong M. Kim in the lab, we isolate the epithelium. We're doing FACS sorting, and you'll note that the majority of the cells are FVF or FOXA2 negative, but we have evidence in a number of different times now that we have both a low and a high population, and we're following this up with looking predominantly by RNA-seq and ATAC-seq and a number of the other different methodologies that your lab is using in terms of epigenomic marks to really try to understand how do these two populations of cells, or really three, how are they different, and then understand gene regulation pathways or gene regulatory networks that are impacted by the acquisition of FOXA2 expression and function. And so we have a nice working model looking at where we're trying to identify both how does FOXA2 turn on and what are its impacts in terms of cell-specific gene regulatory networks. And why is this important? Because as I indicated, during the development of endometrial cancer, particularly aggressive endometrial cancer. So that's it for today. I hope you've enjoyed this odyssey. Probably you know more than you ever wanted to know about the development and function of uterine glands. And I hope that the take-home message today is that they have a critical conserved role in pregnancy establishment. And I look forward to continuing to work on them for the next foreseeable future because glands are cool. So thanks, and I'll be happy to entertain any questions that you have.
Do you have endometrial organ cultures?
that's a good one. So one thing that we have never succeeded is taking the neonatal uterus out and having it develop in vitro. And we have tried 6 ways to Sunday. I mean, we've tried floating it on lens paper. We've tried reducing oxygen, increasing oxygen. Some of the newer cell culture models were using rotating, well, they're really not new, but they've come back in vogue again, which is rotating cultures. We have a mutator that we found on campus that nobody was using. That's great for making assembloids, but it doesn't promote development of the uterus. And so it's really been kind of an intractable project. And so here's what I think is actually the problem. And I didn't get into it today, but so Skip Bartol at Auburn University has a really nice model where he has always studied development of the pig uterus. And what he basically found in a series of studies is that there are milk-borne factors from the mother that are ingested by the neonatal piglet, and these lactocrine-acting factors actually impact development of the uterus. And I think, you know, how many of you have ever looked at like a day 3 or 5 pup? How many raise your hand? A mouse. So, I mean, those little pinkies, right? And what immediately do you see? Like they're chock full of milk. I mean, it's like they're sitting on top of an egg. And I literally think that this is probably one of the single problems that we're missing, is that there are factors coming from the milk that are circulating, and these extrinsic factors are impacting basically the ability of that early uterus to maintain its integrity. Because when we culture them in vitro, basically the luminal epithelium just undergoes apoptosis and disappears. And I think it needs those extrinsic factors. So I have a new graduate student, and we're actually doing that project. We'll see how it works out. So. All right, good.
When I was taking retrovirology, some people consider the fetus as pretty well regulated tumor. So is that fetus being used to see how oncogenes are regulated for treating cancer, for example?
The short answer is I've never heard any of those studies before.
Because there are many oncogenes in the fetus.
Right.
And they play a very important role in duplication replication. However, these many oncogenes are pretty well regulated.
Right.
And I wonder if that form of regulation in the fetus could be applied to regulate tumor.
Yeah, I mean that's a good thought, and I haven't thought about that before. So good. Well, is any trainees or students? Yes, ma'am. That's a very good question. So I think, like when I was a student, like the predominant idea was really reproductive tract secretions were not important for sperm at all. And I think that's because you could do, IVF and ICSI. It is well recognized now that secretions of the oviduct actually are very important for early embryogenesis, that you get a better quality ovary, and essentially, you know, all modern day embryo media is essentially a derivation of synthetic oviductal fluid. Whether or not the uterine glands impact that process, they probably do, but you know, the conventional theory, right, is the sperm that get up the fastest are the ones that undergo capacitation and eventually, you know, fertilize the egg. But that's a really under-investigated area. I think second is we'll talk about the microbiome aspect. That has been highly contentious in the field of reproduction. There were some groups that were isolating, you know, early on with 16S sequencing and, you know, finding signatures in the placenta. And so it turned out that the vast bulk of that was actually contaminants because of the way, I mean, labor and delivery is not exactly the cleanest process on earth. And so I think that has been refuted. Now, when you really look in a normal uterus, there's almost no microbiota present. Now, there are an amazing amount of flora in the vagina and cervix, but particularly the vagina. But most of that never reaches the uterus except for in certain cases where you have a uterine infection. And like in the domestic world, that's predominantly like a retained placenta. You'll get a pretty bad infection in the uterus. But normally the uterus is essentially, you know, there's really no microbiome present in it. Yes. Well, I'm going to take him first and then you. So why doesn't the uterus reject the embryo? Yeah. There's a couple different mechanisms. One is that, you know, the uterus from an immunological perspective is the site for the embryo. And so a lot of the normal immune systems, once those cells enter the uterus, they undergo a phenotypic change where now they're pro-embryo rather than anti-embryo. And some fascinating work done with both T cells as well as natural killer cells, which in the uterus are not killer cells. That's been really some fascinating work that predominantly in the human literature. The other one is that in reality, there is some recognition outside of pregnancy recognition signals that are secreted by the placenta of the embryo. And we talked about yesterday, like how does the endometrium can sense even differences in a female and a male embryo. But the processes that are regulated actually are not anti-embryo. And in only rare cases do you actually even see like class 1 MHC expressed by a normal placenta. You will see it during rejection, which has been shown in nuclear transfer-derived clones, as well as some amazing work done in horses. But generally, the idea is that, you know, the uterus is set up to be pro-pregnancy, not like anti-pregnancy. And progesterone really is the major driver of all that. Yes?
I don't know if you mentioned it or I just missed it, but what exactly, like, do you know what components the uterine glands are secreting? And then have you tried, or is that a possibility to try and sort of exogenously giving those secretion factors in a knockout model and then
Ooh, that is a really good question. And so number one is it has like everything under the sun in it. And we published a lot of different, we did a lot of omics on uterine fluid. Like we had a whole program in extracellular vesicles at one time as well. So you can find pretty much everything in there. There's selective components of serum. There are proteins and micro and macronutrients that are directly synthesized by the epithelium. There are amino acids and sugars like glucose and fructose in there. But the problem is it's like a Sigma catalog experiment, right? Like what is the most important, and without kind of a high throughput readout, like how do you determine what is the most important factor or the most relevant factors? And so we did a lot of what I would call library catalog experiments, but we kind of gave up trying to figure out what are the essential factors. But it still remains a really good question. The second one is, can you do adoptive transfer of uterine fluid from, let's say, a control uterus into a uterine gland knockout? We never published these studies. We tried many different ways to do that experiment. And we did where we would take urine flushings out, concentrate them and directly inject them. Then we did a series of osmotic pump studies where we could take a 2 mil osmotic pump and put a catheter through the oviduct. And we had some success, but we never could get it to be very reproducible. So occasionally we'd see that they would have conceptuses that would survive, and they looked more developed, and in other cases they wouldn't. I think at the time we didn't really have, what the limiting factor was is we would breed the uterine gland knockouts and kind of hope that they were pregnant, right? And we didn't have an in vitro embryo production system, and if we did, then we could have kind of made them pregnant by embryo transfer at the same time that we started putting the uterine fluid into them, but we just weren't really advanced enough to do that at that stage. We also tried it in mice, and that became technically almost impossible. Their uterus, the surgical approach is beyond our abilities. But really good questions. So, yes.
Okay, last question.
Thank you. So thank you so much for the wonderful talk. I just have a question I think can help me build the vision. So when there's a slide, you have a knockout, a sheep has a knockout, don't have gland, and then you mentioned they have early miscarriage. They can have the baby, but they miscarry. It kind of reminds me a lot of friends' story I heard. Their mom spent a long time to carry them, they due to the miscarriage. So can you help me connect the gland problem with the precision medicine that you mentioned?
Right. Yeah, that's a good question. So essentially, it's not, we wouldn't call it miscarriage. Usually in clinical medicine, miscarriage means you actually have a fetus and you have a heartbeat by ultrasound, and then you lose that pregnancy. Oftentimes, at least in humans, the mother will not even know that the fetus has been lost. They won't really know until they start going through menstruation. In the sheep, really, that early blastocyst, which has to undergo in a 2 or 3 day period this elongation, we just found that basically they stayed as a blastocyst, or we couldn't find a blastocyst anymore. But that was very early on. I mean, we're talking like day 10 to 12 of an almost 7-month gestation period. In terms of regenerative medicine or a precision medicine approach, you know, one would be, let's say, could we upregulate FOXA2 in endometriosis and prevent, you know, endometriotic explants from developing in the peritoneal cavity? That's a little bit of a pipe dream, and you never know with CRISPR gene therapy. The other thing is that, so the actual endometrium, you know, in women is about 1 centimeter full thickness during the mid-secretory phase, which is always amazing to me. I mean, your whole life depends on that, right? But women that have thin endometrium have a higher rate of problems with pregnancy establishment, as well as an increased rate of miscarriage. And then there are certain circumstances, particularly Asherman syndrome, where women that have gone through a curettage type abortion, or they have to go through, sometimes you get these things called intrauterine adhesions. And really the treatment for them is you go in and you scoop out the endometrium. And that will, in some cases, prevent adhesions, but also if you remove too much of the lower portion of the basalis, then you don't get regeneration of the endometrium, because that's where the stem cells rise. And so there might be some precision medicine approaches where, you know, if you can infuse, you know, epithelial progenitors along with a stromal cell population, that you could get regrowth of the endometrium or supplant its regeneration to allow for a more functional endometrium that would allow for pregnancy establishment. So, thanks. Now, Wei-Jun said it was the last one,
Okay.
but because you raised your hand and you look nice and you're smiling, sure, no problem.
In your model, your knockout model, did you saw any other lesions in the other departments?
Just in the uterus. And I think it's because, you know, it's really the only part of the female reproductive tract that's not fully formed is the uterus. Now, there's other parts that become more functional, like during puberty, right? But there's, like, you know, they completely lack determined epithelium and then these bifurcated glands. And so I think that's why we're able to overcome that. So, well, thank you very much. I really enjoyed this, and I appreciate all of your time and attention.